Professor Joe Spencer from King's College, London. So you did probably not have a too far journey to come here.
No, I'm on the doorstep.
So, Joe will talk today about human intestinal lymphoid tissue in time and space. Okay, you can see the space already.
Yes, actually I think so as well. I was saying to one of the colleagues, I feel as though I come from another planet anyway because I'm an immunologist. I try and deal with complex systems. I think what we have in common is that we're trying to understand the way human tissues work, whereas you're sort of reducing to look at single parameters. I'm trying to get through the mess of looking at tissues as a whole. So let's see how we do. Okay, so I'm interested mostly in B cells and in human gut. For those of you who aren't familiar, B cells are the cells that circulate through blood and occupy lymphoid tissues. They express a B cell receptor on their surface, and when the B cells are activated, it's that receptor that goes on to comprise the antibodies that regulate the invading pathogens. So I work here in Guy's campus just south of the river in King's College London. And as I say, I'm interested in B cells. So B cell development starts in the bone marrow. Is there
the green, yes.
Yeah. It starts from precursors in the bone marrow, and through this process of gene rearrangement where single V, D and J segments are chosen from multiple alternatives and put together, each B cell emerges from the bone marrow with a unique receptor on its surface. So they emerge as transitional cells. First T1 develop into T2. And we know that this happens in humans because there are therapies that get rid of all the B cells, and so we can observe the order that they come back. It happens in mice this way as well, but we know it also happens in humans. And these cells go on to mature to naive cells. And then the naive cells, if they recognize antigen and require the appropriate help from T cells, they can go on to form this structure called a germinal center. To form a germinal center, which is an area where the cells proliferate like crazy. And as the B cells divide, they mutate the genes that encode the B cell receptor so that the variants of the B cell receptor that are formed can be selected, so only the highest affinity ones go on to leave this process. So you have memory B cells encoding high affinity antibodies, and the plasma cells that secrete high affinity antibodies. And this process of mutation is a very helpful one, as I'll show you later, for trying to understand the history of B cells in people, because it's a sequentially applied process. So the cell type that I'm going to be talking about today is a marginal zone B cell, whereas this scheme is reasonably well accepted. It's not necessarily known where the marginal zone B cells fit into this. So what are marginal zone B cells? Oh no, first of all the processes. So the two different ways that it's been proposed that they develop is one model says that actually this T1, T2 progression occurs after cells leave the bone marrow. They can then become T3 cells marked by this marker CD45RB. And they then mature to marginal zone B cells, and the molecules on their surface are CD27 and IgD. And there is a very good paper supporting this who identified the CD45RB and this axis. For example, this one published in JX Med in 2014 that identified the importance of Notch 2 in that transition. However, there's a whole bunch of other immunologists who don't believe that that's the case. And they say that actually marginal zone B cells are just the earliest emigrants of the germinal center. So they have entered here, they've progressed slightly, but they've not gone through the full process of generating high affinity antibodies. So the question is, does it go like this, or is it simply an early connection to memory cells? So I know that you're not necessarily immunologists, so what? So I think what we're trying to understand here is a very basic feature of human B-cell immunology, how a particular subset of cells emerges, and I think that's important to understand how to vaccinate them. It's important to try and understand lymphomas and immune processes such as those that happen in the gut. So this is the question that we set out to tackle. So what is a marginal zone B cell then? So the term marginal zone was coined by studies of the spleen. So this is a tissue section of human spleen, stained all the B cells in brown. And this area here is the germinal centre that I described, where all the B cells are dividing and accumulating somatic mutations. This section is stained for IgD, so you can see that the germinal centre is surrounded by cells that have high levels of IgD, surrounded by cells that have lower levels, and these are the marginal zone B cells. So this is the phenotype of the cells we're interested in. So they're important in human physiology in that they generate immune responses to bacterial antigens. If you don't have marginal zone B cells, or if you have a splenectomy earlier in life, for example, you find that those individuals become susceptible to infections with bacterial antigens, such as pneumococcal pneumonia, for example. They appear in the first 2 years of life. That suggests that there's a gradual acquisition, so that babies are more susceptible to infections with these bacteria before they're 2 compared to the rest of life. They have somatic mutations in their antibody genes. So as I mentioned very briefly, those somatic mutations are acquired in this structure called the germinal centre after activation. That implies that marginal zone B cells during this first 2 years of life have somewhere acquired these mutations. And lastly, marginal zone B cells in the spleen have circulating counterparts. So that is relevant in humans. Mice don't have them. Actually, I think my next slides tell you what is true of mice. So this is absolutely true of mice as well. So marginal zone B cells in a mouse are responsible for making immune responses to those bacterial antigens. However, if it took a mouse 2 years to generate them, it would probably be dead before it got one. Right? So mice have marginal zone B cells that develop very early on. There's a very clear phenotypic or developmental difference. They don't have mutations in their IgV genes, and they don't migrate. So this is an area where there's a very fundamental species difference, so looking at humans alone is a really important thing to do. So gut is involved in B cell development in many species, and one of the first described areas of the gut that's involved in B cell development was the bursa of Fabricius in chickens. So this was described in 1959, and originally this was where B cell got its B from. It's from the bursa. Now we associate with it with the bone marrow, but actually the B cells were found to be those that came from the chicken bursa. And this structure is very much like the lymphoid tissue that you see in the gut. Secondly, it was shown that in sheep, the lymphoid tissue in the gut is a site where the whole repertoire is developed. So in humans, most of that repertoire, all of the antibodies, all the B cells have got different antibodies on, is generated in the bone marrow. In sheep, that is generated in part in the gut. And most recently, it's been shown that in a rabbit, if you take away most of the lymphoid tissue in the gut, so the rabbit has a massive appendix, if you take away the rabbit's appendix, it no longer is able to generate B cell responses. So based on this then, plus the concept that, sorry, that didn't come out terribly well. It's nicer on my computer, but never mind. So this is, we wondered whether the gut has a role in B cell development in humans. So this is a tissue section of colon, which is normally there on my computer. That'll teach me not to connect my own computer and use the PC. So this is stained for B cells. So you can see that most of the colon doesn't have B cells. This is the lymphoid follicle here that's comprised mostly of B cells. And in the lumen of the gut is the microbiota. So most of the gut epithelium is designed to have a very thick mucus layer. It keeps the bacteria at bay. However, the areas over this dome region of the lymphoid follicles attracts the antigens from the gut lumen in. It can initiate interactions between dendritic cells, T cells and B cells until the B cells start to divide to form these germinal centre structures. So in this microenvironment, there is upregulation of the gut homing integrin, alpha-4 beta-7, and also very high levels of somatic hypermutation are introduced. So these B cells, when they leave the Peyer's patches, can go via the mesenteric lymph node and the lymph. Because they've acquired the homing receptors, when they go to the blood, they can then home back to the gut. So then these cells that have homed back to the gut become antibody-producing plasma cells. They secrete IgA, and the IgA regulates the microbiota in ways that we don't completely understand yet, and it would be good to introduce them into a gut on a chip. So IgA then regulates the microbiota, but at the end of the day, this is an area where there are chronically activated germinal centres from a few weeks of your life. So within a month, a newborn baby will have acquired germinal centres. So it seems logical that if you're going to acquire a cell type that has somatic mutations from very early, that it should involve a site where these structures are found. So with that in mind, we set about studying the lymphoid tissue in humans to try and address this question about where the different cell types are and how they might develop. So to do this, we took gut-associated lymphoid tissue from 8 donors. We took tonsils from 5, and we took 5 spleens. So these are all from different sites. There's the spleen, the tonsil, and 3 different sites in the gut, and I've got that on the slide. And we stained these tissues, so we made single-cell suspensions, and we stained them with this big range of markers for lineage, migration, and function. And then we put them through a piece of equipment called the cytoph. I'm not sure whether you'll be familiar with this, but the cells are coated with many antibodies, and then they're nebulized, and when they go through the argon plasma, they're ionized, and the metal conjugated antibodies are ionized, and the signals from each cell are counted by this time-of-flight mass spectrometer. So you have a signature of the antibodies that are on the surface of each cell. So we acquired the data with bead standards and undertook other quality control, and then performed Intestinal analysis. So we checked that the different tissues would segregate together when they were analysed by multidimensional scaling, and that all seemed reasonably sound, so we were happy that our data is reasonable quality, and we took it from there. So we're not mathematicians, and we don't know what we're doing most of the time, but this in terms of mathematical analysis, but this data, this platform cytobank is very user-friendly, so that biologists like us can input our data and try and visualize it in different ways without requiring much noise. So this Visny analysis, this is taken from the cytobank platform, places the cells together in 3-dimensional space, looking for resemblance between them, and at the end of the day splats them back into 2 dimensions, so that we can then see them on the Tisney plots. So I've got examples of this on the next slide. So what we did here was we took our markers that we know define the B-cell subsets according to the literature, and we asked Cytobank, by that method that I've just shown you, to place the cells in terms of resemblance to each other according to these parameters, and this is what it gave us. So we can see, for example, CD27 is a marker of memory. So where this is hot, there's a lot of CD27 positive cells. Some of these are IgA and some are G, so that's consistent. So this looks fine, but there's not a lot we can do with it, because it's these. You see some plots with some populations that are studied, and these things form discrete islands. But for us, it's all like a solid mass. So another thing that they allow you to do on Cytobank is to generate a spade plot from the Visny. So this is the sort of thing that you would see in a spade, where now it's taken the same data that was on the previous slide and joined cells together by how they most resemble each other. So the things that are in adjacent nodes resemble each other more closely than those in distant nodes. The size of this represents how many cells are there, and this scale represents the intensity of expression of the particular marker that you're looking at. So we then took the data from our B cell analysis, and we generated these spade plots. So we drew these bubbles ourselves. It's not electronic. It's completely manual. We did it. So we chose which bubble every dot went into according to the phenotypes that you would see in the literature that relate to these cells. It was something that the reviewers of our paper had a bit of trouble with, because at the end of the day it's not undirected, and what people want these days is undirected analysis. We didn't do that. However, because we only used a proportion of our markers to generate these things, what we could then do is ask, okay, of these subsets that we have manually defined like this, where are the other markers that we didn't use? And this is the one that relates to the CD45RB, which is the marginal zone precursor cell. And if we take this naive B cell gate here, what we can see is the CD45RB positive cells sit in this corner of the naive B cell gate, and they sit adjacent to the marginal zone B cell population. So for me, this was one of those moments that you dream of in your life when you look at something you don't know what you're going to see, and the two things that you think might be related just sit by side. So a young mathematician that we do have good fortune to work with took those cells, and she did a bootstrap analysis to ask how are they different to the rest of the cells in this gate? Can we call them a separate population? And through this bootstrap analysis, she said, well, yes, they have higher levels of CD24, IgM, CD180, for example. So in that case, we drew a separate bubble around here, and we call these cells their marginal zone precursor, and suggested that they develop in this direction. Which is, if we look at the models, see the two models that I've put together to start with, I think actually we are looking at a different one. So we think from that cytop analysis, it looks as though this branch actually comes from the naive population. However, if we put these two things side by side, this T1 is here, these are the T2, and the naive are in the middle. Then we can see the T3 are close to the marginal zone. Germinal centre, I haven't animated, but memory is way over there. So this is consistent with the model. So I've said that marginal zone B cells are the separate subset that have this reactivity with bacteria. They have marginal zone B cells in tissues have this phenotypic continuum with a potential precursor population, which suggests that they develop through this independent pathway separate from memory. Okay, so that's so far. So I call my talk Space and Time because there is this property of somatic hypermutation does give you the chance to actually look at the sequential acquisition of mutations. So this is a typical structure of B cell receptor of an antibody molecule, and each one is unique in that it's formed by a V, D and a J segment. So this is the gene, and this is spliced to the constant region. That is unique for every cell that's developed, because this is 1 in 50, 1 in 23, 1 in 6, and when they join together, they don't join precisely. There's some non-templated additions. So for every B cell, this is different. So the gene that's inside the cell codes for the antibody on the surface, and we know that the cells then act according to the antibody, but the gene contains the signatures of what went on. So this is a section of human tonsil. So the cells circulating in the blood will enter the lymphoid tissue through the hind epithelial vein. If this cell becomes activated, it will completely change its phenotype as it enters a germinal center response. So this cell can then divide, so it has multiple copies, and it can start to acquire these somatic mutations in the V genes that generate that variability on the V. And it undergoes sequential changes, and it can also, as this is now changed from, so it's not obvious, but it's supposed to change to pink. And then as cells leave the germinal center response, they re-express their antibody, which has now been mutated, tested, and better than it was before. This one has IgD and IgM. and this one is emerging expressing IgA. So processes have happened during this response in a sequential way, and so it's possible to build the lineage trees where it may be that there was a precursor here that you didn't see, but as you acquire somatic mutations, you get this picture of time, that this is how this clone developed. So in our study, because these cells have a different surface molecules to these cells, we can isolate them separately using a cell sorter according to these molecules, but we would know that they're part of the same clone because they've got the same antibody genes in them. So we took samples from the colon, Peyer's patches, and around the appendix. So this covers about 1 metre in distance. And we also took a sample of blood. We took these cells, we made single cell suspensions from these tissues from 3 individuals, and then we amplified the antibody genes using regular PCRs. So for some, we didn't isolate cells by their expression of IgA, but we did use PCR to identify which are the IgA, the IgG, and the IgMs. So these all underwent next generation sequencing, and so now we can have a look at where the clones are, how they've disseminated, and if the same clone is in different parts of the gut and in different populations. So this is a sanity check, quality control type picture. So if this is donor 1 here, this is appendix, colonic follicle, Peyer's patch, and blood. The diagonals are the donor 1 appendix and donor 1 appendix, and the maximum number of sequences are there. But what you can see is the appendix also shared some sequences with the colonic follicle and with the Peyer's patch and the blood. These boxes show the clone spread within a person, and the sparsity out here is consistent with the sharing that you would expect between 2 people. So these numbers are very low because there are some common clones that are common sequences that are shared, but this again is reasonable to suggest that we are looking at dissemination with a person. We're not looking at PCR jumping around our plates, which is always important to eliminate. So this lineage tree shows the dissemination of memory around human gut by aligning these antibody genes and the sequential acquisition of mutations in them. So, for example, this is an IgA clone. It's mostly IgA. Here's an IgG. So we can see that this IgA clone is in the Peyer's patches. There are also some clone members in blood. This brown colour shows that it's in appendix and colonic follicle. But the point is that a disseminated clone tends to be of all 1 isotype, and you can see these memory clones spread through the gut reasonably evenly, suggesting that this happens some way back in time. So it's possible, for example, that somebody who maybe had a childhood infection with a virus expanded these clones, the clones disseminate, become stable and protect through life. This is an IgM clone doing the same thing, so the isotypes tend to remain reasonably separate. So this is memory. This was the data that we were most fascinated to see. So this is a clone of marginal zone B cells and related cells in the germinal centre. So I've said before that the data that we had was consistent with the idea that human marginal zone B cells develop through interactions in gut germinal centres. And here we have cells isolated from gut germinal centres that are clonally related to the marginal zone B cells. Here's an example of marginal zone B cells emerging from a germinal centre, and there are others where they're before. So it looks as though they can circulate, they can enter the germinal centre, they can get mutations and leave. So we're sort of seeing human B cell development through the gut with time. And we can also see clone members in blood, which is consistent with this idea that they can circulate between the guts and the skin. So I've been through this slide before. I think what this work has shown is that the marginal zone B cells acquire through the first 2 years of life, because during that time this population is developing in the germinal centres of the gut, and in that time they're acquiring these somatic mutations in their V genes. How are we doing?
Okay.
Okay, so this one is probably too much for the immunologist, but let's go with it anyway. So here, this is a population of cells that we call IgM only. So they have IgM like marginal zone B cells. They have the CD27, which is indicated of memory, but they don't have IgD. And what we've asked here is how frequently amongst clones do you see the cells that have this phenotype related to the IgA class switch memory cells? And actually you see a reasonable number. So these numbers indicate clone sharing, and in this particular case there was a lot of clone sharing between these cells that we call IgM only and the IgA. And we also see sharing between these IgM only cells, and these are the marginal zone cells. So where these are zeros, in this case we didn't see any, but in these 3 donors, in each case we saw sharing. And there's 9 opportunities for sharing, because we've got cells that are from the same place, but where the IgA has come from one source and the colonic follicle is from another source. So there's 9 opportunities for sharing. And in these 3 of the 4 donors, we saw that. So this suggests that IgM only cells can be related clonally to class switched IgA memory and also marginal zone. However, when we looked at the marginal zone derived clones, we didn't see any relatives in this data set that were class switched to IgA. So, which is consistent with the concept that actually marginal zone B cells and memory B cells, although they can occupy the same space, remain clonally separate. So we propose that the IgM only cells doesn't mean anything in terms of phenotype, and we don't know what they are. Some of them can be clonally related to class switch memory, and some of them can be related to marginal zone, but these 2 are different. So these 2 populations that look like memory, one is memory, one is not. So then marginal zone and memory B cells can both enter Golgi germinal centres and diversify their antibody genes, but they're actually separate things occupying the same microanatomical space. So this, at that point in this study, the BRC that we work with at Guy's got this piece of kit. And we were all rather, as a histologist, I was really excited about it, because this is an imaging system that's related to CyTOF that enables you to visualize in depth in a tissue section. So we were amongst the first to set this up in the UK. So this involves, you have your piece of tissue or cell line, and just like CyTOF, you can stain with a panel of antibodies. Then you ablate the tissue section using an UV laser, and just like with the liquid mass cytometry, you can capture the image by using the mass spectrometry to look at the metals that have been vaporized. So then you can extract the multiple images, and then by data processing you can put them together. So this isn't our data, this is from the catalogue. So in order, what we wanted to do, so we've proposed then that marginal zone B cells come from gut germinal centres. We wanted to see where they are. Can we actually see them? Are they there? If what we've said is true, we should be able to see them. So we used that method with this panel of antibodies that can identify all our B cell subsets using the Hyperion mass cytometry system. So when you use this method, you start off with a cell like a section like this, and you have to place your piece of tissue in the middle, and then you have to tell the people where you want to ablate. So this is a method, they vaporize for you a millimetre at a time. So you have to really know where you want to go and what you want to see, because it costs hundreds of pounds for each millimetre and 4 hours to actually acquire the data. So you can't say, do that section for me please, you have to tell them exactly where to go. So they identify these areas, they ablate them, and then provide you with the imaging data. So this was the first thing that they gave back to us. They stitched these sections, these different areas of ablation together in MATLAB. So we can see the CD20, the B cells are in green, the T cells are in red, pan-keratin's in yellow, IgD is in blue, CD86, which is associated with antigen presentation, in pink, and another interesting marker that's associated with epithelial B cells called FcRL4 in white. So the guys that we are working with specifically stitched together our area of interest, and so we then wanted to see whether we could see the marginal zone B cells by putting them together like this. So the problem that we had is that, so we need, in order to identify marginal zone B cells, we need CD20, which is the pan B cell marker. We need to know that they're IgD positive and IgM positive, though IgM isn't on here. The precursors would be CD45RB, which is blue, and then they have to be CD27. And not all of these markers, so CD45RB is also on T cells, CD27 is on naive T cells. So this actually is just way too complicated to be able to see individual subsets, and the resolution, to be honest, is not great because it's all at a pixel depth. So what we did was to ask the guys in the BRC if they could take the data from this image and generate each pixel as a multi-dimensional piece of data, so that we could treat each pixel as though it was a cell using the platform that we normally use to analyse cells. So the pixels were generated and we uploaded the pixels into the cytobank to analyse in the same way that we'd identify cells. So we then generated one of these Disney plots, and the first problem that you can probably see is that this is for CD20. Most things on here are not B cells. If you look back here, right, so this is our B cell zone, most of things aren't B cells, so it stands to reason that when we actually asked what the pixels were, most of them are B cells. So we then made the spade plot, which didn't look like anything we'd made before, and dragged all of the dots that had the signature of B cells and put a separate bubble around those, and then did a second VISNI on here, and now we can start to see that there are areas of B cells representing different subsets. So now we could manually pick out where, in the same way that we manually picked the other site off, we could now start to take out what we thought were our subsets and then place these subsets back onto the image so we can see where our pixel groups are located. This is an example. This is the marginal zone B cell pixels in green. So this is a sanity check. So we know that the germinal centre should be red and that the CD27 IgD positive cells should be surrounding the germinal centre. So that all looked fine. So we then asked where our unknown populations were. So what we can find here is that the marginal zone B cells are in green. They do indeed locate very close to the germinal centre, and the memory B cells form this peripheral zone around the outside of the B cell area. Again, I suppose it's logical with the incoming antigens would come from the outside, that memory B cells should be located on the periphery. Okay, so marginal zone B cells then occupy this niche between the germinal centre and the follicle-associated epithelium in the gut, and they're surrounded by classical memory cells. So I guess the take-home message is that humans are really more like rabbits in this respect. So it seems as though the gut of a rabbit in terms of B cell development is more like the gut of a human than any other species. Sheep are a bit like that in that the Peyer's patches support B cell development, but in a slightly different way. We're a little bit like chickens, where the B cells are dependent on the gut, but not really like mice. And I think it's the same. Google image. In fact, I showed my daughter this, and she said that's a rat, but you called it musculosis.
Yeah.
Okay, so if I can just acknowledge my collaborators on that work, I've got a little bit more work just to show you on ulcerative colitis before I finish. So this is all published. It was in Nature Communications at the end of last year. Most of it was done by Yuan and Tom and Jacqueline, who's our mathematician colleague from Cambridge. I've got a long-term collaborator, Anna Rosenkamper at Queenary, who's been studying B cells together for a while. These are the guys who had fabulous BRC, and it was real fun working through the imaging mass cytometry with them. Brian helped us with the next generation sequencing, and Max was the person who first described the marginal zone precursor and brought that question to us, so we're very grateful to him. Steve and Mohammed at Yale developed the, worked through the pipelines for the clone analysis of B-cell receptor clones, and these are all our wonderful clinical collaborators who help us with supply of tissues. Okay, so from there, one of the reasons I'm going through this is that I'm now going to just show you unpublished work, and I didn't really want